everyone for coming. No clapping is necessary. Thank you all for coming. It's really fun for me to get the chance to talk about nitrogen fixation in the tropics while I'm in the tropics. And after thinking about it for a while, I realized my title wasn't appropriate at all. And what I really want to discuss today is how much nitrogen can a nitrogen fixer fix if a nitrogen fixer can fix nitrogen. So that's just a joke. So why do we even care about biogeochemistry? As ecosystem ecologists, we like to think about processes that emerge at broad spatial scales. So for example, how much NPP does this temperate forest support? But oftentimes, what's more important is what's happening below the ground at a very small spatial scale. So for example, how much nitrogen these ectomycorrhizal roots are able to capture from the soil and then translate that back to NPP. Well, how and why do these constraints arise in the first place? What are some of the mechanisms that organisms employ to overcome these constraints? So plants have evolved these fascinating symbioses with fungi and bacteria to capture the nutrients that limit their growth. And what you see here are two different scenarios. The solid line is a standard global vegetation model that looks at carbon. So what this is doing is modeling the basic physiological process of photosynthesis. The tropical biogeochemistry has long been considered a very sort of uniform, phosphorus-limited biome. And I would like to sort of take a step back and think about how we consider the biogeochemical underpinnings across biomes. And so if we go all the way from Arctic to boreal and then to temperate and tropical biomes, we see two general patterns. First, we see a decline in the amount of organic matter that accumulates in soils, and we see an opposite pattern in the amount of nitrogen fixation that these ecosystems support. Now, these two patterns have an effect on a number of other processes. For example, the type of nitrogen that is most abundant in those systems and the type of nitrogen that plants rely upon. And there's also a shift from predominantly nitrogen-limited ecosystems to predominantly phosphorus-limited ecosystems. And so this is why we need to understand nitrogen fixation in the tropics. What controls fixation and what potentially limits fixation. So today I'm going to talk about free-living nitrogen fixation and symbiotic fixation. And for both of these, I want to present a new framework to consider biogeochemical constraints. So first I'm going to start with free-living bacteria that are nitrogen fixers. These are asymbiotic. They don't form symbiosis with trees. They are heterotrophs. And then I'll talk about nitrogen fixation with legumes. So these free-living heterotrophs prefer carbon-rich, nitrogen-poor soils. And that makes sense because they're heterotrophs, they need the carbon in order to get energy, and their function is sort of irrelevant in nitrogen-rich systems. So this is the niche that we'd expect to see them in, leaf litter and decaying organic matter. And he started this project where we used the Gigante fertilization plots that Joe Wright has set up in order to measure the amount of fixation that's going on. Now, remember that this is nitrogen fixation by these bacteria. This is what's happening in the leaf litter. So he measured fixation in control plots and in the nitrogen addition and phosphorus addition plots. And when you look at this, it's really not that surprising, the pattern that you see. The addition of nitrogen suppresses fixation. So why fix nitrogen if you can get it for free? The addition of phosphorus stimulated fixation. And this sort of goes along with how we think about tropical systems. They're thought to be phosphorus limited. But what was really curious was that there was a similar positive response to the micronutrient treatment. So it's kind of strange to think of two different elements. So maybe one of the micronutrient elements and phosphorus both limiting fixation at the same sort of level. So why do we even care about these other elements when we're talking about nitrogen fixation? Now, the reaction itself is very costly in ATP. 16 molecules of ATP are required per molecule of dinitrogen that is reduced. But I think, and there's, of course, there's phosphorus in ATP, but I think the importance of ATP has been sort of inflated, and I think where phosphorus is really important for nitrogen fixers is if you're an organism and you're fixing nitrogen, you need to maintain stoichiometric balance. So for every unit of N that you fix, you need to take up also from the environment phosphorus in order to balance that and maintain stoichiometric balance. So clearly phosphorus is important. Now molybdenum is interesting because there's only one atom, this red sphere right here, one atom of molybdenum in each of those cofactors. But this enzyme cannot function without that molybdenum. So in this entire thing, we have two atoms of molybdenum represented. And one way to think about this is the organisms that can fix probably don't even express the gene to synthesize this protein unless molybdenum is already present. So as Alex was pondering the results of those data, he stumbled upon some information about phosphorus fertilizer. And this sort of changed everything about his project and also what a few of us have also focused on. It turns out that triple superphosphate, which is the most commonly used fertilizer, especially in the tropics for fertilization studies, is contaminated by trace amounts of Mo. This changed the interpretation of our previous results. The simplest explanation for this pattern is that molybdenum that was purposely added in the micronutrient treatment and molybdenum that was unintentionally added in the phosphorus treatment was stimulating nitrogen fixation. We added molybdenum, we saw a beautiful positive response. And then we added phosphorus and we saw no response. We even added phosphorus at along 3 orders of magnitude, just to make sure we weren't adding too much or too little. The other point to take away from this is that all of those previous experiments with phosphorus fertilizer need to be completely rethought. So every experiment that I know of in the field with phosphorus fertilizer in the tropics has used triple superphosphate. And it's really difficult to go back and say whether. And so now this is just one site, and how can we interpret this across the tropics in general? And so you remember how I told you that at the beginning that tropical forests are considered to be phosphorus limiting, but it's clearly not as simple as that. This is total soil phosphorus in parts per million in the top 20 centimeters of soil across a number of tropical sites. And we have soils that are almost as high as 2,000 ppm in Hawaii, Costa Rica, and down to about 40 parts per million in the Guiana Shield region of the Amazon Basin, as well as Borneo and Cameroon. And so what we have are 6 sites that are in mature forests. These are either secondary mature or old growth mature. And we have a range of total phosphorus from about 100 to 700 or so, although there is an area in the AVA soil type that's as high as 1,800 parts per million. And here we're thinking about total phosphorus as simply an index of ecosystem level phosphorus status. So these sites are within BCNM or immediately south. This is the low phosphorus site Rio Paha on the rhyolitic tuff. And the reason for these strong differences in soil phosphorus is related to the complex geology of the region, where we have this rhyolitic tuff and marine sediment parent material developing into relatively phosphorus poor soils, and we see andesite and basaltic parent materials giving rise to phosphorus rich soils. And so our question was, do phosphorus, molybdenum, or both elements limit fixation? And how does this limitation, or does it, change does the element that limits fixation change along this landscape gradient, soil phosphorus? So we visited each of these sites and we did the same small scale incubations where we added analytical grade phosphate that had no contamination with molybdenum. We added sodium molybdate at a reasonable dose, and then we did both of them together. And we compared that to control that just received water. A positive response to both Mo and P together, but not to those two elements individually, only when they were together. And at the high end of our gradient, we saw a positive response to molybdenum alone in 2 out of the 3 sites. And so to summarize these results, not every site responded to nitrogen additions, and I suppose if they did, that would have been far too simple. But the point is that when a limitation arose, it followed this pattern of molybdenum and phosphorus co-limitation in P-poor soils, and molybdenum only limiting fixation in phosphorus-rich soils. So we can now think about these systems with a little bit differently. We know that there are these really strong vertical gradients in soils where phosphorus is very abundant in the upper soil and then it becomes less abundant with depth, whereas molybdenum seems to do the opposite. And this seems to be driving the element that's limiting fixation. But we also need to be thinking about broader patterns in soils. And so along our landscape gradient soil phosphorus, we saw a shift in molybdenum and phosphorus co-limitation in P-poor soils to molybdenum only limitation in P-rich soils. With all this new knowledge that we have about P and Mo and landscape gradients and phosphorus, we then wanted to understand how nitrogen, phosphorus, and molybdenum may control nitrogen fixation in the form of symbiosis. So to start with, I conducted a greenhouse experiment with Sarah Batterman, a PhD student at Princeton. She was leading this work, and we did an experiment where we had 3 levels of nitrogen additions by 3 levels of phosphorus additions with this nitrogen-fixing mimnosoid, Ginga punctata. What about molybdenum? So in that other experiment, we gave all the other essential elements that the plant would need. We just wanted to focus on N and P. But it's also interesting to know how P and Mo relate to one another in constraining nitrogen fixation. Okay, so so the greenhouse study demonstrated to us that phosphorus is a key driver for fixation. Nitrogen, of course, is suppressing fixation, and that at high amounts of phosphorus, or perhaps when plants are growing a lot, molybdenum can also limit fixation. So to summarize all of this, we have seen evidence that both nitrogen, phosphorus, and molybdenum can all control fixation to a certain degree. N is a suppressor, P acts to limit, and molybdenum can also co-limit. And so last, this also fits into our new conceptual framework for how nutrients are controlling fixation with depth and along landscapes. We see shifts in the elements that limit fixation for free living fixers and for symbiotic fixers. Thank you, everyone.